Okay, the last thing we get into is DNA analysis. So we know DNA structure, we know each person has a specific sequence of nucleotides, and that's what makes them them. It's what makes them unique. So on DNA analysis, it's a way to identify who a body sample, for example, blood, belongs to. We're going to do that by looking at the DNA and seeing how the DNA differs from somebody else's DNA. So here's your 5 major steps, your big picture. The first step is to get your sample. When we get our sample, we're talking about DNA extraction. So when you extracted out the strawberry DNA and our own DNA, you saw that DNA. We extracted it away from the cell because we only want to analyze the DNA. We don't need the cell membranes. We don't need the nuclear membrane. We don't need the organelles. We just need the DNA. So we get our sample first. Once we have just DNA, we run it through PCR. PCR is polymerase chain reaction. What that does is it amplifies the amount of DNA. Amplify means makes more. So we're going to go from probably thousands of copies of DNA to millions of copies of DNA. And it only takes about 40 minutes. We need lots and lots of copies. That way we have enough in order to see our results. So we get our sample, we run PCR. Third thing we do is we add restriction enzymes. And restriction enzymes are going to cut the DNA into fragments, so into pieces. Now, at that point, we just have a solution that just looks like liquid, and all it is DNA in tons of little pieces. But we can't see the DNA in pieces. We still can't identify it. So you have to think in the real world when we do this, we don't know we trust that all of this is happening, but for getting our sample and PCR and adding the enzymes, we can't see anything. We just hope it's going on. It just looks like a little vial of liquid. But when we run our gel electrophoresis, it's going to separate out those DNA fragments based on 2 factors. We'll get to that. And then finally, we interpret the results and we see who the DNA belongs to. So a couple key things. First thing is the restriction enzymes. So remember, big picture. We get our DNA sample, we run PCR, then we add restriction enzymes. By running restriction enzymes, an enzyme is just a protein that has a job. So a restriction enzyme's job is to search the DNA for a specific sequence and cut it. So what we used when we were in class is the restriction enzyme HEY3. We said it looks for the sequence GGCC. But when you look at GGCC, that's not double-sided DNA. It's just one side. So you have to fill in the other side. So it would be CC, G, G. So this is the sequence that we look for, and it cuts it right down the middle. So if I look at my fragment here, okay, so if I'm following along, we have GGA, TTC, ACT, GGCC. So there it is. So what I do is I box it out so I can identify it, and then we keep going, AAGCTTGG. So there's no more places to cut, but the restriction enzyme is going to cut right here in the middle. So it's going to take this fragment and it's going to make one cut. If it makes one cut, that means that there are 2 fragments left over, one right here and one right here. Now, the next thing we do is we count up how big are those fragments, and you count the number of base pairs. So when we count the number of base pairs, we go 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. So this side's 11 base pairs long. This one is 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So that one is 10 base pairs long. Now, the key thing to remember about restriction enzymes is they search the DNA for a specific sequence and they make the cut. Now, here's one thing I don't want you to forget. When you're looking, you look at the double-sided DNA. So we look for GGCC, and the bottom is going to be CCGG. So we have GGCC, the bottom is CCGG. You only have to look along the top line for GGCC. You don't come back and look at the bottom line, because you have to remember DNA is double-stranded. So if you search the top line, the bottom line is going to be opposite of it. You don't have to search it again. So you only look at the top line for GGCC. You find your cut, and that's where you know the DNA is going to make the cut. Now, remember, in the real world, we don't know where it's cutting. We can't tell because we just see a vial of liquid. So this is just to illustrate so you know what's actually happening on a molecular level. Okay, so our fourth step is gel electrophoresis. Now, gel electrophoresis separates out the little fragments of DNA we just created based on two things. First thing is we're going to separate it out based on charge. So DNA is negatively charged. DNA is negatively charged. So if you think that opposites attract, the negative DNA is going to be attracted towards the positive pole when we turn the electricity on. So DNA is a negative charge. You cannot forget that. DNA is a negative charge. So it separates it out based on charge. The second thing is based on the size. Now, gel is kind of like a filter. It's like a big thing of, it's like a slab of Jell-O that we run the DNA through, and it's going to filter out the size of the fragments. So the larger fragments are going to get stuck back by the wells. The smaller fragments are going to travel further. Now, a key thing is that fragments of the same size, so same size fragments, will travel the exact same distance. Fragments of the same size will travel the same distance. So if we look at the process of DNA here, remember that DNA is negatively charged. It's a key thing to remember. So we start out. This is our gel, which is just a slab of a Jell-O-like substance. You'll notice that we have a negative side over here, we have a positive side over here, because it's gel electrophoresis, which means we're going to use electricity to separate it out, the positive and the negative. So we always put the DNA on the negative end, because it's going to travel towards the positive end. Remember, opposites attract, negative and positive. So we're going to add the DNA into these little wells here. Then we're going to turn on the electricity, and when that happens, the DNA is going to move through the gel towards the positive electrode. So it's going to move. Then you see here how it separates out a little bit more, and it keeps moving. So the fragments that end up close by the wells are our bigger fragments. The fragments that are clear down here are smaller fragments. This is what it actually looks like, this blue thing right here. That's what a real gel will look like, and you'll do that second semester. But you can see bigger fragments get stuck behind, smaller fragments move further through the gel. So again, let's go over it again. We have our wells down here where we add our DNA sample. We have our negative end. We have our positive end. DNA is going to travel towards the positive end because it is a negatively charged molecule. So DNA is going to travel towards the positive. So we're going to turn on the electricity. When we turn on the electricity, we end up separating out the DNA. The bigger fragments get stuck. The smaller fragments are going to move the furthest because they can get through the filter. So you have big and you have small fragments. Remember, fragments of the same size travel the same distance. Fragments of the same size travel the same distance. So you end up with a completed gel here. Okay, so if we look here, last step is to interpret gels. So recap. We got our sample. We ran PCR to get more copies of the DNA. Then we added our restriction enzymes that cut the DNA to pieces. We put them in the gel, and the pieces, the fragments, are going to get separated out. So the last step is for us to interpret these gels. Again, look, the DNA was put on the negative side right here, so it could travel towards the positive. But here we have 4 different samples, lanes 1, 2, 3, and 4. And the question is, what 2 samples are from the same person? Key thing is, if you have the same banding pattern, you are from the same individual. So if you look here, I'll give you a second. Which 2 are the same? If you answered lane 1 and lane 2 have the same banding pattern, so they must be the same person, you are correct. Lane 1 and lane 2 have the same banding pattern. Now, if you think about that, if they have the same DNA sequence, because they're the same person, then the restriction enzymes would cut in the same spot, which means the fragments are the same size, And those sizes, if fragments are the same size, they travel the same distance in the gel. So these two fragments traveled the same distance, so they landed there. These two traveled the same distance, these two traveled, and those two traveled the same distance. Lane 3 and lane 4 do not have a matching banding pattern, so they are not from the same person. Okay, so in this one, it says who does the crime scene blood belong to? So here we have our crime scene blood, suspect 1, suspect 2, and suspect 3. So again, you're looking for the same banding pattern. So take a look. If you said suspect number 2, you are correct. Because if you compare the banding patterns for the crime scene and suspect 2, they are the same banding pattern because the DNA came from the same person. This banding pattern is also known as your DNA profile. That is your banding pattern based on where the restriction enzymes cut your DNA. Okay, so on this one, I give you kind of a different scenario. We start out here and we have our standards. Now, the standards are what we get from the lab, and the lab tells us, okay, it's going to produce this many bands. So here we have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So it said there's going to be 10 bands produced in this standard. And the 10 bands, band number 1 is 500, and it's 500 base pairs. I just didn't have space to write it. So 500 BP stands for base pairs. The second one is 450 base pairs. And then we get squished here, so I couldn't say what they are. This one right here is 320, 275, 250, and then 100. So they told us how big these fragments are, and that's what we order from the lab. Then we have 4 different samples here. We have sample 1, 2, 3, and 4. So our question says estimate the size of the 3 bands in lane 2. So I'm only looking at lane 2. Okay? So the first thing is I look at this first band and I got to see where does it fit on the standard. So if I take my band and I come over, I know it's below 450. So that means it must be smaller than 450. So it's probably around 430. So about 430 base pairs. Then I come down here and I come over. This one is in between 250 and 100. So I'd say it's probably about 200. And then this one is right above 100, so probably about 110. So if it's 430, 200, and then about 130, we estimated the size by using the standard markers over here. Remember, the standard markers, if it's above the marker, that means it's going to be bigger. If it's below the marker, it's going to be smaller. And see how these ones are pretty much the same size right here? Pretty much in the same space, so they would be the same size. So this one's probably about 450. So you look at your standards and you can estimate the size of the bands. Okay, that's your basis for electrophoresis. Key thing is you have to know how restriction enzymes work to find out how many cuts are made, how many fragments are made. Then you have to be able to analyze a gel. Can you tell me the size of a fragment? Can you tell me who it belongs to based on the same banding pattern that you're looking for? Okay, those are the key things to be able to take away from gel electrophoresis.